Thank you everyone for being here. Today we have the pleasure of welcoming Dr. Gabriel Sawakuchi. He is a principal investigator working at MD Anderson Medical Physics. And on a personal note, I was this close to doing my PhD with Gabriel so many years ago. Did I make the right choice? I'll let the audience decide, but I'm definitely interested to hear about what he's been working on here. So with that, I'll hand it over to you, Gabriel, to talk to us today. Thanks.
All right. Yeah. Thanks, Pete. Yeah. So I was in Canada for 3 years. I was a faculty member at Carleton in the physics department there. So I remember visiting McGill and giving a seminar there. I think it was in 2011 or so. So that was a long time ago. I was just talking to Pete. It's like over 10 years, which is hard to believe. But I have some ties with Canada because or very strong ties with Canada because my older son, he's Canadian. I'm originally from Brazil, and my wife, she's originally from New Zealand, and we live now in the US. But my son, actually, if anyone asks where he's from, he says that, very proud, I'm from Canada. So we still have connections because of he identifies himself as being Canadian, even though he only lived there for 2 years. But anyway, let's get to the talk here. So I'll be talking about. I'm very aware that you guys are working on DART. So I choose that topic because that's one of the projects we have in here. So we've been working with Alpha Tau for some time, and recently have funds from NIH to also do some research with alpha particles. So that will be the focus of this presentation. And okay, so just then some disclosures that I have funds with alpha tau, and the top of this presentation will be related to research that we're doing with them. So just to give a summary of what we do in our lab. So basically, we wanted to understand how to combat cancers that do not qualify for surgery and or are not responsive to therapies. And many cancers are not does not qualify for surgery. Including pancreatic cancer, that about 80% of the patients does not qualify for surgery, either because of metastatic disease or because patients, they don't have a good outcome for surgery. Also, several cancers do not respond or develop resistance to chemotherapy or molecular target therapies, and also radiotherapy. We know that there are several tumors that are radio-resistant, and are also hard to target because they're close to critical structures. And that includes glioblastoma, pancreatic cancer, some lung cancers, and many others. And some or several tumors or cancers also do not respond to immunotherapy or develop resistance to immunotherapy. And this is mainly the focus of this presentation, is how to make these tumors that do not respond to immunotherapy be responsive to immunotherapy. So then the focus of our lab is to develop these new ways to overcome resistance to therapies by understanding the molecular consequences of radiation damage. So then this is just an overview of the projects that we have in the lab. Again, working with alpha particles is one of them, but we have many other projects going on there. I'm showing this because if anyone has any interest in any of these topics or are doing any research in any of these topics, feel free to contact that. We would be really happy to collaborate with anyone that's interested. So we have a project that we're modulating metabolism to sensitize radio-resistant types of tumors. And we're going to be focused on alpha particles to stimulate the immune system. So you're going to be hearing about that on this presentation. We have also focused on trying to come up with molecular signatures that could predict the tumor response to protons, but also to photons. So it's how to predict how if a tumor is radio-resistant or radio-sensitive, just based on the biology of the tumor mutation mainly. And we've been focused for quite a while on using molecular target therapies like DNA repair inhibitors to radiosensitize tumors as well. So then jumping on to the topic of this presentation there's this problem that there are cold tumors that does not respond to immunotherapy. As we know, immunotherapy has revolutionized how we treat cancer patients. But just recently we're finding out that the response rate remains relatively low because of these tumors that does not respond to immunotherapy that we call cold tumors, or also develop resistance to immunotherapy after a certain time. Then we also know that radiation is historically known to produce suppressive immune effects, but very recently several studies demonstrated that radiation can also stimulate anti-tumor immune response that could be used to sensitize immunologically cold tumors to immunotherapy. And because of that, it became very important in our field, radiobiology and radiation oncology, to understand what are the mechanisms in which radiation stimulate this anti-tumor immune response. And this is to maximize the use of radiation to sensitize tumors to immunotherapy. So then I'm going to just briefly describe a little bit of how we target tumors with immunotherapy. So the most common way to do it is using immune checkpoint blockade. So immune checkpoints are cell surface proteins that regulate the immune system. And this immune checkpoint proteins, they're expressed in immune cells, normal cells, cancer cells, and what they do is just to inhibit the activation of T cells to prevent T cells from killing health cells. But cancer cells, they're very good on hiding from the immune system because they usually express these immune checkpoint proteins. So immune checkpoint blockade just breaks or prevents these checkpoint proteins from binding with their partner proteins, which releases the brakes of the immune system to then allow the immune system to recognize that this is a cancer cell and then kill the cancer cell. Then one of the pathways that's very important in immune stimulation is this cGAS pathway. So cGAS is an enzyme that recognizes and binds to double-stranded DNA. So any double-stranded DNA that's foreign to the cell or is in a place in the cell that should not be is recognized by cGAS, or meaning in the cytoplasm. So if there is the DNA from a virus in the cytoplasm of the cell, cGAS can recognize this and activate immune response. But also any nuclear DNA in the cytoplasm will also activate this pathway. And that's when it starts to have a link with radiation, because radiation will generate double-stranded DNA in the cytoplasm. And this was recently also discovered and many simultaneously. So these two papers were published back to back in the same issue of Nature, and you can see that the page numbers here, they are back to back, and this was published by independent groups. Where what happens that micronuclei So micronuclei are small nuclear-like structures, and you can see here on this image. Get the pointer here. You can see on this image here. So the blue is the nucleus, and the micronuclei are those nuclei-like features here that they have a nuclear membrane and have fragmented chromosomes inside of them. So radiation can induce micronuclei formation, and micronuclei form following mitosis after failed DNA repair. So micronuclei is also surrogate for persistent DNA damage. Okay. So radiation then can induce this micronuclei formation when where now this micronuclei is floating in the cytoplasm. When the membrane of this micronuclei ruptures, it spills DNA or double-stranded DNA to the cytoplasm. That this now is recognized by cGAS and then activates a cascade of events to stimulate the immune system. So what our interest is to find out, how we can maximize the production of micronuclei, and if there is a type of radiation that will generate more micronuclei than other types of radiation. So then now I'm going to talk a little bit about physics. So ionization density, as we probably are all aware, is how radiation deposits the energy spatially. And we know that there are types of radiation that have high ionization density or high linear energy transfer, and types of radiation that has low linear energy transfer. So high LT radiation, they're densely ionizing, so they deposit the energy very, very packed. And this is a very, very nice figure from the radiobiology book of Eric Hall, where simulated tracks with different LTs in different types of particles are superimposed to a micrograph of a cell. So this is the nucleus of a cell, just to have an idea of the dimension of the events of energy deposition. But as you can see, high LT, in this case, is a very low energy protons, have very, very densely ionizing events, where when we start thinking about the dimensions of biological targets like DNA, the probability of causing damage is much higher for high LT than compared to low LT. And I'll talk a little bit more about this later. So as I discussed before, just some definitions here on this slide of LT and comparing LT with dose. So dose is the amount of energy, average amount of energy deposited per unit mass, while the LT is how that energy is distributed spatially. So more or less describes how the energy deposit is distributed spatially. And as you know, there are types of radiation that are used clinically that have higher LT than photons, and we are familiar now in our field with proton therapy and also with carbon ions. So those type of particles, they can have not necessarily, but they can have high LT. Then low LT particles, this is it's more or less arbitrary, and this is what I put in here, but there is no border or threshold between low and high LT. But I'm defining here clinical photons or high energy protons, so protons at the entrance or at the skin of the patient, who have an LT of about up to 2 keV per micron, depending on the initial energy of the beam. Then high LT will be protons at the tumor, where the LT will vary from about 3 to up to 12, 5 to up to 12 keV per micron. Carbon ions at the entrance will have an LT of about 13 to 20 kV per micron, and carbon ions at the tumor will have an average LT of about 50 to 90 kV per micron. Alpha particles will have an LT of around 70 to 200 kV per microns. So then, coming back now to the biology and putting things in terms of the dimensions of biological targets, to understand what's the probability of causing damage by different types of particles with different LTs. So low LT radiation or low ionization density, they are more sparsely ionizing. And most of the damage to DNA is caused indirectly. And this is because this low LT radiation will produce reactive oxygen species that can travel or diffuse to DNA and then damage the DNA. For high LT, because the ionization density is much higher, the probability of direct damage happening to the DNA is much, much higher. So if high LT, we see a lot of direct damage. So in this situation, we also know that this classical radiobiology, that these tumors that are hypoxic, they are less dependent on the radiation with high LT. But also high LT produces more complex and clustered DNA lesions that are harder to repair. So complex DNA lesions are lesions which have different types of damage, but close to each other. So there's base damage, double strand breaks, and single strand breaks, all packed close together. So you can have clusters of all types of damage, but you can also have clusters of double strand breaks. And high LT radiation has the probability of producing these more complex and clustered lesions. Then the next question is how the cells will cope with radiation-induced damage. So low LT induces more simpler lesions that are easier to repair. And the cells would repair these many double strand breaks that they are more detrimental and lethal through repair pathway that's called non-homogeneous joining. And non-homogeneous joining is by far the most important double strand repair pathway. High LT will induce, as I said, these more complex and clustered lesions that are harder to repair. And then there's another double strand repair pathway that the name of it is homologous recombination, that would be able to cope with these complex and clustered lesions. But it's still most of the damage is repaired by non-homogeneous joining. So as the LT increases, the contribution of homologous recombination to the repair of the double strand breaks increase, but still by far the main repair pathway is non-homogeneous joining. So we've been studying this in our lab for some time, and we published a paper to show that that's the case. So if you want to know more details about that, take a look on this paper that was worked by Scott Wright from my lab. So as I mentioned, so then LT may affect the presence of micronuclei. And this data, unpublished data, this shows that a higher LT radiation generates more micronuclei, and this is the if you look here, so I'm going to explain this figure here because it's very packed. So these are cells that were exposed to either not exposed or exposed to true grays of 6-MeV X-rays or true grays of highly T protons. And this is measuring the number of micronuclei. So it's basically counting the number of these features for each of the cells. And we can see that the higher the T, the more micronuclear induction per nucleus we're gonna get. But the other thing that is important to consider is that micronuclei formation is related to the ability of the cell to repair the DNA damage. So then is also related to how effective a cell can repair that damage. So it's also related to DNA repair. And here we can show that by inhibiting DNA repair. So we use a drug that can inhibit a very specific protein that's related to homology recombination repair. And we can also see an impact on micronuclei production. So there are many ways to modulate the number of micronuclei production by radiation. One is by modulating the LAT. The other one is by modulating the DNA repair as well. So then our hypothesis is then that high LAT would induce more complex and clustered DNA lesions that are hard to repair and resulting in the formation of micronuclei and consequent activation of the cGAS immune response pathway. And I'm going to try to show that this is some data indicating that this is the case now. So then some sources of high LAT radiation, just to put things into context, that we use in clinical radiation oncology. So as I mentioned before, proton beams is something that's becoming more and more popular because the technology is getting cheaper, but it's still very expensive. So with protons, the tumor receives a dose average LAT of about 3 to 12, 3 to 7 kV per micron. But for really high LAT protons, with a LAT of higher than 50 kV per micron, the volume of the tumor exposed is very, very small because a 50 kV per micron proton has a range of only 5 microns. So helium ions as well. It's also very expensive, and there's only one place using helium beams to treat cancer patients, and this is in a clinical trial. But also the volume of the tumor that would receive LATs of higher than 50 kV per micron is pretty small. And carbon ions, that's super expensive. But that would be something that would be comparable to alpha particles, where the LAT is about 50 to 90 kV per micron on the tumor. So sorry, this is not right. So 50 kV per, this is a mistake on these slides. So a 50 kV per micron carbon ion would probably have a range of a centimeter or so, I think. So then other alternative sources of high LAT radiation includes radiopharmaceuticals that are alpha emitters. And there is a FDA-approved alpha emitter to treat metastatic castration-resistant prostate cancer. So the nice thing about radiopharmaceuticals is that we can target them. We can use conjugate them with antibodies that will target very specific surface proteins in cancer cells. And then they can also be delivered systemically to treat metastases. So this field is currently booming. So there are many pharma companies, including very big pharma companies, that are developing and testing alpha emitters. in several clinical trials. So this field is getting a lot of attention now. And as you guys know, we can also deliver high energy radiation with diffusing alpha emitters radiation therapy. That's alpha DART. So alpha DART is a local therapy, and we have to implant seeds in the tumor, and it's based on the decay of radium-224 to radium-220. And then radon is a gas that diffuses into the tumor up to 2 to 3 millimeters away from the source. And this overcomes the very short range of alpha particles. So alpha particles have a range of no more than 100 micron in water. So to be able to deliver it locally, we have to overcome that limitation of the range. So in this way, the range of the alpha particle is not limited by the range itself of the alpha particle, but it's how far radon diffuses in tissue. So in that way, we can still have implant decent number of a number of seeds to cover the tumor, but not having the whole tumor covered with seeds. So that's where I'm going to be focusing from now on. So first, to be able to understand how alpha particles, the radiobiology of alpha particles, we first build an alpha particle irradiator to be able to do experiments with cells, in vitro experiments. So we use an americium 241 source that's very, very high activity. So it's a 5 by 5 cm source, and then we build a custom cell dish to be able to expose the cells to this source. So this cell dish, that's what's important, and there are a lot of technical details on that, and I'm happy to, if anyone is interested in building alpha particle, happy to discuss the technical details about it. But basically, the cells need to be on a very, very thin layer of material, because the alpha particles, otherwise the alpha particles stop before reaching the cells. So we use a mylar, a 3 micron thick mylar sheet to cover the cells on this mylar sheet to be able to expose it. We have the americium source, and then the dish, or the bottom of the dish, is made of mylar. The cells are adhered on this dish and on this mylar, and then the alpha particles comes from passing the mylar and reaching the cells. So we characterize this irradiator using topaz, and this was the work from Dr. Perlis that is collaborating with on this research, where he simulated the exact setup that we use to expose the cells. And this is some parameters of the irradiator. So how the dose ratio is a function of depth. So depth here is from after the downstream the mylar surface. So this is inside of the medium, or the water, on this photo here. So the mylar surface is here, the depth dose 0 is at the surface. So you can see that the dose varies, or the dose rate or dose varies quite rapidly as a function of depth, where the penetration of these alpha particles is only about 14, 15 micrometers. So in that setup, at the surface, the average LET is about 180, and at 5 microns is about 190 or 194. Then we also characterize the profile of the source, or the profile in general, and is very flat. So we were happy that this was not a heterogeneous field, so we would be able to give homogeneous dose to our dish. So then to calibrate this or to determine the dose rates of the source, what we did, we use EBT3 films, but this is a different EBT3 film. Is unlaminated. So then the sensitive detector, part of the detector, is exposed to air. So then alpha particles can actually reach the sensitive area of the detectors. So we took a bunch of ABT films, sent it to our collaborator at Oxford, Dr. Hill. So they have a very well characterized alpha particle irradiator there that they know the energy of the alpha particles, the LET in different conditions, or they also calibrated to those in the different conditions. So we send a bunch of detectors to them and ask them to expose them with different doses in different LETs of alpha particles, and then we got those detectors back to then calibrate our source. So we used two methods. One was using topaz and also using this unlaminated EBT3 films, and we got very good agreement. So before then I start showing some of the biology data on the source, I just wanted to highlight a lot of challenges and limitations of alpha particle experiments. And this is mainly due to the limited range of alpha particles. So first, the cells, they must be adhered to the mylar. If they're floating in the media, the cells that are floating will not receive any alpha particle. So this will bias quite a lot the results, because you think that the alpha particle is exposing the cells, but the cells are just there in the media that's not getting any alpha particles. Also, even if the cells adhere, the cells cannot pile on the top of each other because alpha particles do not reach a cell that's on the top of another one because of the limited range. Also, not all cells will work, because some cells, they don't have a flat morphology, so they're more rounded, and in that way the nucleus can be far enough such that we don't get any alpha particles on the nucleus. So the distance from the surface of the mylar to the nucleus needs to be small. Also, the cells must have a thin nucleus to minimize partial volume irradiation, because again, these alpha particles are stopping at around 15 microns. The LET will change inside of the nucleus as well, and also the dose will change inside of the nucleus as well. And also because cells have slightly different morphologies, even for the same cell line, the doses will be different for every cell. And these differences will be even higher for different cell lines that will have, by definition, different morphologies. So it's really hard to compare apples to apples here then, because by definition, if you have two different cell lines that they have different morphologies, they're going to be getting different doses. But then we have no other choice. That's the only way we can expose or have a high throughput way to expose cells to alpha particles. So we're able to validate our setup and workflow with some cell lines. One of them is this murine cell line of pancreatic cancer, where the cell line behaves pretty well in mylar, and so we can work with this cell line quite well. So this is the chronogenic cell survival. So is the survival factor as a function of dose. cells of this panel 2 cell that was exposed to X-rays and to our alpha irradiator, just to show that we have like a more linear on the log scale, more linear survival curve. That's high LT, one of the consequences of high LT, and also a very high RB. So we're happy when we were able to get these results, because then we're now very confident that our setup works. So Eurico was a student from Portugal that spent some time in the lab, and he was the one acquiring this data. But this is to show that this is also well known in classical radiobiology, that IDT generates more DNA damage and more persistent DNA damage. That's what's important for us here. So what we did, we exposed again cells to X-rays and alpha particles, the same dose, and compare them. And so we look at time points that was 4 hours after irradiation, 24 hours after irradiation, and 72 hours after irradiation. So right after irradiation, about 4 hours right after irradiation, the number of gamma-8 tracks, that's N50.1. So those two proteins are surrogate for double strand breaks. So right after irradiation, the number of double strand breaks for alpha particles is lower actually than for photons, and this is mainly because the fluence of alpha particles is much lower than the fluence of photons for the same dose. So there are many less hits in the nucleus of alpha particles than for photons. But the important parameter is how the cells can cope with this damage. So then after giving the cells some time to repair, the cells will repair much more of the X-ray induced DNA damage than the alpha particle damage. And that's what's important. You know, what's the residual DNA damage there, or persistent DNA damage. And here we can see at 24 hours and 72 hours that alpha particles they can induce much more persistent DNA damage. And the consequence of that we already discussed is the formation of micronuclei. And again, this illustrates that alpha particles at again larger time, a long time after radiation, can induce much more micronuclei than photons. So then this is a potential application for this is more immunoactivation, as I discussed before. Now moving on to animal studies to show then we can also show a consequence on immune stimulation and T-cell activation. We need to be able to do this experiment with alpha particles in animals. And again, it's challenging because of the limited range of alpha particles in tissue. So we can do either using radiopharmaceuticals and deliver systemically. So there are several radiopharmaceuticals that are available in our alpha emitters that are being tested in clinical trials. This could be done as well with several suppliers, but it's very challenging to use radiopharmaceuticals in the lab because of radiation protection issues. And again, we're going to be using Alpha Dart for that. So how we get our Alpha Dart sources? We get them from Alpha Tau. They're directly shipped from Israel to us, and there's a lot of synchronization that needs to be done to be able to do these animal experiments. because the half-life of the sources are only 3.6 days. So we need to time such that we load the right activity at the manufacturer of the source, and we have at the day that the sources arrive, or the day of implantation, we have the tumors on the right tumor size on the mice, in the mice. So we need to really optimize the workflow here and understand how after inoculation of the tumor cells, how long it takes to grow, and have all these details figured out. So there's sources with several sizes that can be loaded with different activities. So we can also play with this parameter to see how the tumors respond. In several applicator designs, then the applicators, they can already sterilize with preloaded sources in them. So it's very easy to use. So the first thing we wanted to do when we started doing this is to just come up with a workflow from ordering, receiving, doing some quality control, and implanting the sources into mice. But also testing the different sizes of the sources with different activities and what will be the response of the tumors. So we started with this very aggressive triple negative breast cancer model in biopsy mice. So here is just some illustrations on the applicator and how we implant the sources. In this case, the model is the tumor is in the leg of the mice. So we had these two sources, dimensions, one with 5 millimeters, the other one with 10, and with different activities. One was half of the other, but the activity per length was the same. And then we did some tumor growth delay experiments where we measure how the implantation of the source would affect the growth of the tumor. So as a control, we use inert sources, so source that was not loaded with activity, but it was exactly the same as the active sources, just to make sure that we have a proper control. And then we saw that both 5 and 10 millimeter sources delayed tumor growth, but the 10 millimeter sources were much more effective due to mainly the higher activity. Then we also wanted to look if there is any just by itself, if alpha particles will have an ability to induce some immune activation. So we measure this ratio between CD8 T cells and Tregs. So this is one of the parameters that is used to that indicates an immune activation, the ratio between CD8 T cells and Tregs. And we saw that alpha particles induce an activation compared to the inert seeds. So we also are trying to develop different models, and we demonstrated this also for this aggressive pancreatic cancer model. That's the KPC cell line, where in this case was a different size of the seed as well. There's a 6.5 millimeter alpha source with the same activity as the 10 millimeters one that we use for the breast tumor model. So these experiments are still ongoing. So we just started these experiments a few weeks ago, and so we're going to have more results in here. But again, the main point here is that we now have two models that we can work with. One breast cancer model and the other one pancreatic cancer model. Then the main question is if alpha particles can sensitize tumors through immune checkpoint blockade. So with this experiment, what we wanted to do is to test two things. If indeed alpha particles will sensitize ICB, but also if our workflow was robust enough. So we wanted to repeat this several times to make sure that we get the same result. Again, this needs to be done in synchronization with alpha tau. So we have sources with the same activity. The size of the tumors in the animals needs to be similar if we do in different months this as well. So everything should be consistent, but we want to check if all those parameters affect the response. But also the schedule of drug, the schedule of CTLA-4 that was the immune checkpoint blockade that we use in this case. So first we confirmed that for these 3 experiments that was performed in a period of about 6 to a year, 6 months or 12 months. So they were not done together, but they're done within the span of a year or so. So the consistency of the activity during the day of the implantation was pretty good. So the tumor sizes of these 3 experiments were the first and the second were similar, but then the third, the tumor size was a little bit higher than the other 2. But we saw very, very good consistency between those 3 experiments. So here on the top is just the alpha DART, not with the anti-CTLA-4. And we see that among these 3 experiments, the tumor growth delay is very, very consistent. So there's no statistical significance difference between the tumor growth delay on these three independent experiments. And when we treat with the CTLA-4 as well, even though we use different schedules of the CTLA-4, it seems that the schedules that we use is not very sensitive to the response that we get. So we're happy with that. And then we went to compare what's the effect of the CTLA-4 and did a large experiment with many cohorts. So first we focusing on this figure here, A on the left, we measure tumor growth delay for different cohorts where we have only the inert source, then the inert source treated with the immune checkpoint blockade, then DART with DART only. So the IgG is just the control for the anti-CTLA-4, and then with everything in it. So the DART plus the anti-CTLA-4. So one thing to notice here that's very important is that this tumor is immunologically cold. It does not respond to immune checkpoint blockade by itself. So if immune checkpoint blockade is delivered just by itself, there's no difference in tumor growth delay. So the immune checkpoint blockade will not do anything with this tumor. So this tumor is very immunologically cold. Then if we treat with alpha particles, we see a bit of tumor growth delay compared to the controls or to the control or to the anti-CTLA-4 alone. But then when we combine both, we see that now DART can sensitize the immune checkpoint blockade, where this should be related to an immune stimulation. And as you can see here, we can see significant differences in the tumor size on day 14. And also significant difference on survival as well. So then here we did show data indicating that we might have an immune stimulation. But to actually directly show this, we need to analyze the tumor microenvironment. That's the work that we're doing right now. We don't have that data yet. But with that, we do have some other indications that would more or less indicate that there is some immune activation that's also systemic. So from those mice that we euthanized after the end of the experiment, we collected tissue, we collected the lungs and look for metastases in the lungs. So just looking at the number of metastases, we showed a trend, but we didn't see a large difference. And we also score other things, including tissue disorganizations. Is a score that Dr. Marinello, she's an instructor in my lab, but she has a background in pathology. So she came up with this to analyze alterations in the tissue parenchyma. That's also related to the metastasis. And we also see a trend, but not significant. But looking at inflammatory cells, so if we get the regions of metastasis and within those regions, we look where the inflammatory cells are, we see that when we have the cohort with DART plus NCTLA4, we have a significant difference on number of inflammatory cells, which is, again, is an indication of some systemic immune stimulation, but until we directly prove it, we're going to have to do some more analysis of these inflammatory cells to confirm that these are T cells and they are activated T cells as well. And then the other experience that we did that is related to immune activation as well is to look at how we can use the repair inhibitor to enhance even more the effectiveness of alpha DART to sensitize immune checkpoint blockade. But this is also important in the field of cancer, because of PARP inhibitors are being widely used now and are FDA approved. But there are many tumors that do not respond to PARP inhibitors or develop resistance to PARP inhibitors. And this shows that alpha DART can sensitize alpha inhibitors as well. So this is data where we use two different PARP inhibitors. One is talazoparib, that's FDA approved. So that's the BMN673. The other one is inhibitor it's a very new inhibitor that's in clinical trials now, and it's more specific to PARP1 from AstraZeneca. And the inhibitors, just the PARP inhibitors by themselves alone, they do not control the tumor. So you can see here. But then when you combine them with DART, DART can sensitize them. So this is also important because it could be that with the combination of DART plus the repair inhibitors and immune checkpoint blockade, you're going to get even better immune responses. So then just to summarize things, alpha particles have high relative biological effectiveness, and this mainly resulted from inducing much more clustered DNA lesions that are harder to repair. So consequently, alpha particles will induce more micronuclei and potentially causing more cGAS activation and anti-tumor immune stimulation than photons. Some tumors are resistant to radiation, and alpha DART delay tumor growth in aggressive triple-negative breast cancer model and also in aggressive pancreatic cancer model. Some tumors that are immunologically cold and not respond to immune checkpoint blockade, but alpha DART can sensitize those tumors to then respond to immune checkpoint blockade. And likely, this is through the ability of alpha particles to activate T cells. On the PARP side, also some tumors are resistant to PARP inhibition, but alpha DART can sensitize tumor that is resistant to PARP inhibition. And localized dose of alpha DART may help to minimize immune suppressive effects of radiation. that also when combined with DNA repair inhibitors can minimize toxicity to normal tissue. But there are many questions we have, and this is some of the questions we're trying to answer now. So is how alpha particles activate the immune system? And these are questions that everyone that is doing immunology research applied to radiation oncology applied to immunology is also trying to answer these questions. So is how radiation activate the immune system, in particular alpha particles in our case. There are many other mechanisms of activation. It could be through immunogenic cell death, other mechanisms of sensing RNA instead of DNA. High DT can create maybe more binding sites for cGAS because you're going to have more double-stranded DNA fragments with high DT. Also, how DNA repair inhibitors modulate the alpha particles response of cells to amplify the anti-tumor immune response. And again, one of the important things that must happen for cGAS thing to happen is cGAS needs to bind to a double-stranded DNA fragment. But if the binding site is already covered by a protein that's trying to do DNA repair, then that binding site will not be available for cGAS binding, and the pathway will not be activated. So we're combining different DNA repair inhibitors with the mindset of cleaning those double-stranded double-stranded DNA fragments, such that cGAS can bind to them and activate the cGAS thing. Also, we're trying to understand how alpha DaRT modulates tumor infiltrating lymphocytes, memory T cells, and activated systemic and sustained anti-tumor responses. And try to understand what mutations would be predictive of the response of alpha DaRT to amplify immune stimulation. And the other big question we have is how we can compare alpha DaRT with external linear or low LET radiation because of the very different dose distributions. So I think with that I finished, and I just want to acknowledge all the members of my lab that were the ones that generated all of this data. The folks on Alpha Tau that they've been collaborating very closely, mainly Vered, with us, helping us with the models, teaching us on how to implant these sources in animals, and also the funding agencies. And I think I stop here. Yeah, thanks.
Great. Super. Thank you very much for a very interesting talk. We have time for some questions for Dr. Sawakuchi. Feel free to ask them out loud, or if you type them into the chat, I will read it out loud for you. If anyone has any. I wrote down a few questions, more about my ignorance in radiobiology. It's been a while since I took a course in this. Right from the start, you talked about looking at LET, high LET, low LET, and the effect of, or the relative proportion of double-strand breaks being repaired by non-homologous end joining versus homologous rejoining. Is it well known the differences between the two in terms of their ability to not have errors? Like, is that already a known issue of what is causing more cell death versus the other?
Yes and no. So it's well known that non-homologous end joining would might cause the DNA not to be repaired properly, and introduces like mutations that lead the cells to survive, but also can lead to cell kill. Homology recombination is a DNA repair pathway that will always repair the cells perfectly, the DNA perfectly.
Right.
So if homology combination is the DNA repair that repairs that specific break, the cells will repair. But when you start increasing the LET, it gets to the point where the damage is so complicated that no DNA repair pathway will be able to cope with that, and the cells will die anyway. But on that intermediate range where there is a lesion that's a clustered lesion, if modular combination is repairing, then the cells will be able to cope with that. If homology combination, non-homology joining is repairing, the cells might survive or might have some genomic instability, which will survive for some generations, but then will die. And because we'll activate some mutations that would also induce some mechanisms of cell death.
Okay.
Not
All
sure
right.
if I answered your question.
Well, I guess I was thinking too simplistically, like NHEJ doesn't do as good a job as HR, but then when it comes to actually having the cell die, it's a little bit more. It depends on how bad, like as you said, in high LET versus low LET, it depends on how much of a mess has the radiation made on the DNA. How complicated are the double strand breaks?
Yeah, and the other thing important to remember here is that for immune stimulation, we don't want to kill the cell. For cGAS activation, we want to create some sort of genomic instability where the cells can still live for several generations with high number of micronuclei.
Right.
Because that's what would trigger the activation of the pathway. So if we kill the cell at first, then we're not inducing that pathway. So there's a very fine line there between which doses we need to use, which LET we need to use.
Okay, thank you. Other questions from the Zoom, from the room? If not, I have one more question before we let you go. So you did mention that Alpha DART is in clinical trials, correct, with Alpha Tau?
Yes, yes.
Is there enough patient data to compare, like everything you've done is in animal models or simulation, is there enough patient data to kind of make sense of what you've seen so far? Does it kind of go along with what patient data is showing in terms of, I guess, tumor control and so on?
No, I think the only clinical trial that was already finished was the one at Sloan Memorial Kettering, and that was only for very superficial lesions, and was a safety trial only.
Yeah.
So you guys are doing things more complicated now with pancreatic cancer, but I think you only treated a couple of patients or so, right? So I'm not sure if there's.
I don't know if there's anyone on this meeting.
At the JGH.
Yeah.
He's talking about the patients treated by Dr. Corey Miller for pancreatic cancer, but I think he only did maybe five patients now, no, maybe three to five. So he recently showed his data at one of the conferences, so it was really great to see. He did some really great work.
And it would be nice to know if what sort of biospecimens we're collecting for collecting blood that could be used to analyze some cytokines to see if there is any immune stimulation, because that will be the data needed to like in terms of the focus of my research to understand if a high LET would stimulate immune response more than low LET.
Okay, super. Thank you. Let's see, one question. Oh, Francine Donnell also kind of added in that 8 patients were treated with Dr. Miller at the JGH.
Okay, great.
Yes. So, super. All right, if there are no other questions, I want to thank our speaker again. Thank you so much for talking to us today, Gabriel, and thank you all for being here today, and we'll see you all at the next Friday noon talk.
Yeah, thanks everyone. Thank you for the invite.